Good afternoon to everybody. Thank you for coming. And it is a great pleasure for me to introduce Kim Chuan for this conference. Kim Chuan is a very young researcher that got his bachelor's degree in biological sciences at the China Agricultural University in Beijing, and a PhD in biology at the University of Iowa in the United States. Then he joins Mark Anderson's laboratory in 2015 at Johns Hopkins University as a research associate, and was recently promoted to assistant professor of medicine. At Johns Hopkins, his main focus is on the roles of CaMKII oxidation. Because of his background in basic biology, he has a keen interest in the evolution and physiological functions of CaMKII oxidation, which had only been known for its pathological roles. By looking at CaMKII oxidation from this angle, he discovered that CaMKII oxidation plays an important role in promoting the evolutionary fitness of organisms while conferring detrimental consequences as a trade-off. His work suggests that CaMKII oxidation plays a role in aging, and it might be a promising target for intervention that promote healthy aging. The title of the talk is Evolutionary Physiology, Oxidation of CaMKII, a treaty between God and the devil. So welcome, Kim Chuan. Thank you for accepting our invitation, and we are ready for listening to you.
Thank you very much, Dr. Martiezzi. That was a very kind introduction, and I'm truly honored to be invited to present my work in front of this great audience. So let me share my screen. So today, I'm going to talk about this work, which was recently published in Nature Communications, but I still think it is very exciting to share it with you. So I'm going to tell you this story about how an ancient kinase, CaMKII, gained the ability to transduce signals from reactive oxygen species, and how such an ability is a mixed blessing that both improves physiological performance and contributes to aging and diseases. I have nothing to disclose. So talking about aging, we have to talk about Tithonus. Tithonus was a famous character in Greek mythology. He was a prince of Troy and was the lover of the goddess Eos, the goddess of the dawn. As a young man, Tithonus was handsome and strong, but he was still human. Being aware of his mortal life, Eos begged Zeus to grant Tithonus immortality. Zeus, in a good mood, granted Eos her wish and thus made Tithonus immortal. Tragically, Eos forgot to ask Zeus to grant Tithonus perpetual usefulness. Tithonus, unable to die, nevertheless got very old. It was said that when the loathsome old age presses fall upon him and he could not move nor lift his limbs, this seemed to her in her heart the best counsel. She led him in a room and put to the shining doors. There he babbles endlessly and no more has strength at all, such as once he had in his supple limbs. In fact, aging-associated diseases increases their incidence exponentially with increasing age. With his immortal life, Tithonus likely had all of these problems, such as atherosclerosis, hypertension, diabetes, Alzheimer's disease, and Parkinson's disease. If not, he was still very frail. The tragedy of Tithonus leads to this question: how does our body lose its usefulness when the loathsome old age presses on us? How do we get old? The most influential theory about how our body deteriorates during aging is the free radical theory of aging. It was proposed by D. Harman in 1956. It suggests that the cellular metabolic activity produces byproduct free radicals, in particular, the reactive oxygen species, ROS. These highly reactive chemicals damage cellular constituents and cause aging. Since its proposal, the free radical theory of aging has become a widely accepted theory for how aging happens. Harman's original paper has received close to 10,000 citations. However, as the research community gained more tools to measure and manipulate free radicals in organisms, the validity of the theory has been questioned. Some have even proclaimed that the free radical theory of aging is dead. However, most researchers believe that ROS plays an important yet nuanced role in aging. One of the most important realizations is that ROS are not always blunt damaging chemicals. Instead, ROS induces a hormesis dose response. The effects of ROS can be both beneficial and harmful, depending on the dose of the exposure. And the optimal biological outcome is achieved within an intermediate level of ROS. The hormesis effect of ROS is achieved through signaling pathways that are sensitive to them. These ROS-sensing proteins can regulate a range of biological functions, such as metabolism, growth, survival, and proliferation. Based on the idea that ROS function in large part through signaling proteins, the revised free radical theory of aging suggests that dysregulation of ROS-sensitive signaling proteins is a major avenue by which ROS cause aging. One such protein that plays important roles in mediating ROS-related signals and cause disease is the calcium calmodulin-dependent protein kinase 2, CaMKII. CaMKII is a ubiquitously expressed protein kinase that plays diverse roles, such as enabling learning and memory, regulating muscle performance, heart rate, cardiac contractility, insulin secretion, activation of fertilized eggs, immunity, and more. CaMKII has a kinase domain and a regulatory domain that blocks this kinase domain by preventing it from accessing substrates. It's activated by the rise of cellular calcium, which complexes with calmodulin, and calmodulin in turn binds the regulatory domain of CaMKII, releasing its inhibition on the kinase domain, thus leads to CaMKII activation. In addition to this mode of calcium calmodulin dependent activation, CaMKII is also activated by post-translational modifications on its regulatory domain. In 2008, the Mark Anderson lab identified a pair of methionine residues in the regulatory domain of CaMKII, and methionines are sensitive to oxidation. So we found that oxidation of this pair of methionines by ROS prevents the regulatory domain from blocking the kinase domain, leading to calcium calmodulin independent activity. Because ROS is well known to play roles in various diseases, since the discovery of CaMKII activation by oxidation, for more than a decade, the Anderson lab and others have engaged in studies demonstrating the detrimental roles of CaMKII oxidation in various diseases with a component of excessive ROS. In short, we have shown that CaMKII oxidation is involved in atrial fibrillation, sudden cardiac death in the setting of diabetes, asthma, stroke, cardiac arrhythmia, and cancer. And when we use the knock-in strategy to replace the methionine pair of CaMKII delta isoform with the ROS-resistant valines, the animals were protected from these diseases. So by now, you may have noticed that most of these diseases are aging-associated. However, our mouse studies were done in young animals. How do we know that CaMKII oxidation actually plays a role in aging? Aging is very hard to study in mice. In particular, there are 4 CaMKII isoforms in mice. All are oxidation sensitive. It will be a daunting task to create knock-in mice in each of those based on each of those individual genes. And it will be even harder if we try to create quadruple knocking mice. So we resorted to a different animal model, the fruit flies. It turns out that the flies only have one CaMKII gene, and they have much shorter lifespan. Thus, it is easier to genetically manipulate flies and to study aging with them. Interestingly, we found that flies do not have the oxidizable residues that enable CaMKII to be activated by ROS. Instead, in place of the MN residues, the fly CaMKII naturally have the VV residues. You may still remember that in our mouse experiments I introduced in the previous slide, we used the VV residues to make CaMKII in mice insensitive to ROS. So to take advantage of flies, we used CRISPR to replace the VV residues in fly CaMKII with the MN residues, thereby making humanized flies. So we first tested whether inserting the methionine pair would make the fly CaMKII sensitive to ROS. We did a paraquat sensitivity assay with the flies. Paraquat is a chemical that induces ROS production in cells. We found that flies harboring the MN version of CaMKII are significantly more sensitive to paraquat. And showing these graphs, after paraquat treatment, significantly more MN flies died than VV flies. Thus, changing the VV residues of fly CaMKII into MN indeed makes the CaMKII sensitive to ROS and confers an effect that can be observed with intact animals. To test whether CaMKII oxidation promotes aging, we carried out lifespan experiments with the wild type, where VV, and the mutant humanized MN flies. Male MN flies lived significantly shorter. Our additional experiments showed that there is no difference in size or fecundity between the genotypes during the young age, suggesting that the MN flies are healthy. These observations lead to an intriguing question. Why do we human and mice have oxidation-sensitive CaMKII? Why hasn't evolution weeded out this detriment healthier and longer? After all, flies do not have the CaMKII oxidation, and they do just fine. And the mutant mice that we created to have no CaMKII oxidation in CaMKII delta not only appear to be healthy, but are protected against the ROS-dependent diseases. So wouldn't it be better if we humans have no CaMKII oxidation so that we can suffer less from aging? To answer this question, we need to go back to ask a more general question. Why do we age? We are the products of evolution, a process where genes responsible for deleterious traits are under negative selective pressure and get weeded out with time. It is clear that lifespan is an evolutionary trait that has very strong genetic underpinning. In the labs, lifespan of animals can be extended significantly through artificial selection. And it is also well known that longevity and healthy aging run in families in humans. Furthermore, living longer without aging-associated deterioration is clearly a beneficial adaptation. We can think about Tithonus. What if he didn't age? How many baby Tithonuses he could produce with a goddess wife? The genes that keep him immortal and perpetually young would certainly take a hold in the population's gene pool and become the predominant alleles. So why doesn't evolution make us live longer and healthier? Well, the problem is that there's no free lunch in evolution. Many traits are compromises that increase fitness in some aspects, but also decrease it in others. Aging, in fact, can be explained by the compromise between young and old ages. The most prominent evolutionary theory of aging is the antagonistic pleiotropy theory of aging. proposed by George Williams in 1957. Antagonistic pleiotropy means opposing effects of the same genes. This theory suggests that genes that are harmful at an old age can be retained in evolution if the beneficial rules at a younger age. The reason is that the force of natural selection is weaker on older animals than on younger ones. This is because the chance that an animal is still alive reduces with time, even if the animal is intrinsically immortal, because it can die from accidents, toxin, predation, infectious diseases, and parasites. Therefore, there are fewer chances for natural selection to act on older animals than on younger ones. This decreasing force of natural selection with age allows genes that play beneficial roles at younger ages to be selected for, even if they are harmful during the old age. The antagonistic pleiotropy theory of aging has received strong empirical support since its proposal. Because CaMKII oxidation plays such detrimental roles in aging, we hypothesized that oxidation of CaMKII is an antagonistic pleiotropy phenomenon. In other words, given the role of CaMKII oxidation in promoting aging-related diseases, there must be benefits of CaMKII oxidation that have yet to be discovered. To support this hypothesis, we must provide two lines of evidence. First, we must show that CaMKII oxidation promotes aging and aging-associated diseases. By now, I hope I have convinced you that this is the case. Second, we must show that CaMKII oxidation has important functions that improve evolutionary fitness of animals. Here, we face a problem because CaMKII isozymes are expressed in almost all cell types, and they have diverse functions. It is conceivable that some of these functions rely on oxidation of CaMKII, but many may not. So how can we find a physiological function that might require CaMKII oxidation? To solve this problem, we resorted to an evolutionary approach with the help of Gabe Bever, an evolutionary biologist here at Hopkins. We reasoned that by comparing species with CaMKII oxidation to those without it, we may find clues regarding why CaMKII oxidation evolved. We compared all CaMKII sequences available in the genome database represented by the organisms on this phylogenetic tree, and the result was striking. We found that all vertebrate animals, including fish, amphibian, reptiles, birds, and mammals, have oxidizable CaMKII. On the other hand, none of the invertebrate animals have it. This analysis suggests that oxidative activation of CaMKII evolved in the common ancestors of all vertebrate animals about 650 million years ago, and shows that CaMKII oxidation has been absolutely conserved since its emergence. The results suggest that CaMKII oxidation likely played important roles for the evolutionary fitness of vertebrate lineage. One important and unique feature of the ancestral vertebrates was that they have vastly improved motor function. This increased motor function required more powerful muscles, and thus evolutionary innovation that promoted muscle function was likely favored by natural selection. CaMKII is well known to regulate muscle performance. So we reason that one of the reasons for the evolution of CaMKII oxidation was to improve the physiological performance of ancestral vertebrates. So how can we test our hypothesis, given that the ancestral vertebrates lived 650 million years ago and are no longer in existence? Fortunately, we can first turn back to modern animals. Because oxidative activation of CaMKII likely plays the same roles in skeletal muscle in modern animals, we generated knock-in mice that have their MM residues of CaMKII gamma replaced by the VV residues. We modified CaMKII gamma because it is the predominant isoform expressed in the skeletal muscles. And we also showed that modifying CaMKII gamma doesn't change the relative expression levels among the 4 CaMKII isoforms in the skeletal muscles. Therefore, our CaMKII knocking mice, the VV mice, lose the ability to oxidize the predominant isoform of CaMKII in the skeletal muscles. We then performed a series of experiments on the mice from intact animals to in situ muscle stimulation and force measurement, down to isolated skeletal muscles for calcium handling and real-time CaMKII activity measurement. So in short, our results support the concept that muscle contraction generates ROS, in part through NADPH oxidase in the muscle fibers. And the ROS activates CaMKII through oxidizing the methionine residues. We demonstrated this by using a novel fluorescent CaMKII activity reporter that we developed. And then the activated CaMKII in turn enhances calcium handling in the muscle fibers, which make the muscle more resistant to fatigue, as shown by the in situ contraction measurement. In the end, oxidation of CaMKII in skeletal muscles results in animals that have better running performance, as measured by treadmill running. Thus, our results support the hypothesis that CaMKII oxidation confers physiological benefits favored by natural selection. However, we did these experiments in a modern species. How do we know that CaMKII oxidation was actually evolved to promote motor function in the ancestral vertebrates 650 million years ago? To solve this problem, we again used a technique from the evolutionary biology field. We used the technique called extant phylogenetic bracketing. The logic of this technique can be explained by this evolutionary tree. At the top of the tree, we find the organisms that are alive today or extant. Going down the tree, we find organisms of the past. The branching points of the tree are the shared ancestors of the branches. The extant bracketing technique suggests that if you find that a function is shared between 2 extant organisms, such as A and B, on 2 separate branches of the tree, then the parsimonious hypothesis is that this function must exist in the common ancestors of these 2 organisms, C, unless you find evidence to show otherwise. We now have 2 animal models on hand, the flies and the mice. These 2 extant organisms form a bracket that share a common ancestor deep in evolution, and the ancestral vertebrates were its descendant. We now know that CaMKII oxidation promotes motor function in mice. If it does the same thing in the flies, we can conclude that it could play the same role in the common ancestor of flies and mice, and therefore the subsequent ancestral vertebrates. We tested the motor performance of MM and VV flies with this climbing assay. In this assay, the flies were dislodged to the bottom of these tubes. They would instinctively climb up as an escape reflex. We measured the velocity of the climbing and found that the MM flies climbed significantly faster. Thus, CaMKII can also improve motor function of flies. Furthermore, when we treated the flies with the antioxidant N-acetylcysteine, NAC, the performance of the MM flies was significantly reduced, whereas that of the VV flies was not affected, further suggesting that the performance benefits of CaMKII in the flies requires oxidation. To determine if CaMKII plays a role intrinsic to the muscles in flies, we analyzed the contractile performance of fly hearts. This was done by Mira in Tony Camarado's lab here at Hopkins. The fly hearts is a muscular tube that can beat spontaneously. We isolated the hearts and performed high-speed imaging and analyzed the performance of the heart. We found that the hearts contracted and relaxed more quickly, consistent with improved performance. Again, when we treated the hearts with the antioxidant NAC, the performance benefits of hearts was reduced, while that of the VV hearts was not. The results suggest that the performance benefits of having residues in CaMKII requires physiological levels of ROS. Thus, we completed the extant phylogenetic bracket and showed that CaMKII oxidation likely conferred performance benefits in the ancestral vertebrates, which might be one of the reasons that it was fixed by evolution in the vertebrate lineage, despite its detrimental effects in aging. By now, we have both lines of evidence to support that CaMKII oxidation is a molecular example of the antagonistic pleiotropy theory of aging. We further showed that the role of CaMKII oxidation is not limited to improving motor performance. We did a transcriptomics profiling with muscles from and VV mice kept sedentary or ran for 350 meters. We found that exercise altered the transcriptome of mice more strongly than the VV mice. The mice are the ones with CaMKII oxidation in the muscles. So as shown by this principal component analysis, you can see that the transcriptome shifted more in the mice after exercise than the VV mice. To further determine what biological pathways are most influenced by CaMKII oxidation in the muscles in response to exercise, we did an ingenuity pathway analysis. We found that CaMKII oxidation is required for the transient inflammatory response to exercise in skeletal muscles. This observation is very interesting because inflammation also plays important roles in aging. It suggests that the detrimental roles of OX-CaMKII might be related to its role in promoting inflammation, particularly in the context of chronic activation of CaMKII in the aged animals. We then further extended the gene regulation roles to the oxidation of another CaMKII isoform, the CaMKII delta, in a different cell type, the mast cells. I compiled a list of about 90 candidate genes that might be regulated by mast cell degranulation, based on the fact that they are regulated by CaMKII gamma in muscles, and that they are reported to be regulated by mast cell degranulation in human in a previous study. With the help of our collaborators, Peisong Gao and Dando and Hopkins, I showed that mast cell degranulation induced a stronger transcriptional response in the presence of CaMKII delta oxidation, and shown by this principal component analysis, where the transcriptional profile of mast cells shifted more strongly than the VV mast cells. Both inflammation and mast cell degranulation are part of immune activities. Our data thus suggests that CaMKII oxidation plays a wide range of physiological functions, and many are waiting discovery. I also found that CaMKII oxidation plays a similar role in regulating gene expression in the flies, which is not shown here. Thus far, our data make a strong case that CaMKII oxidation is an exemplar example for antagonistic pleiotropy theory of aging. Specifically, we showed that oxidation of CaMKII can boost physiological performance in both vertebrates and in invertebrate animals, and that it promotes aging and aging-associated diseases by transducing ROS signals. We further validated the detrimental roles of CaMKII oxidation in the flies. In the previous slide, I showed that oxidation of CaMKII promotes motor performance and physiological levels of ROS. And This effect was abolished when we treated the flies with the antioxidant to quench the ROS. To test the role of CaMKII oxidation under elevated levels of ROS, we fed the flies with 4 millimolar paraquat for 24 hours. This treatment significantly reduced the climbing performance of the MM flies, while the VV flies were strikingly unaffected. We then treated the flies with even lower dose of paraquat, 1 millimolar, and monitored their motor activity for 6 days with the help of Ian Blum in Dr. Mark Wu's lab at Hopkins. Showing in this graph is the daily ambulatory activity of the flies. The solid lines up here are the flies that are treated by control food, and the dotted lines are the flies that are treated by food tainted with paraquat. As you can see, the VV flies here are not affected by paraquat treatment for their motor activity, their ambulatory spontaneous activity, whereas the MM flies after treatment had significantly reduced motor function, even by this low-dose paraquat treatment. Thus, we think CaMKII oxidation may also contribute to frailty, which is shown in aging. To further assess the involvement of muscles directly, we again performed cardiac performance analysis, as I showed before. In this case, in the presence or absence of paraquat. As shown here, the MM hearts performance are significantly suppressed by paraquat treatment, both for contraction and relaxation, whereas the VV hearts are not affected. When we treated the hearts with paraquat for longer duration, we found that many more MM hearts became dysfunctional, while the VV hearts are more resistant to paraquat. The examples of VV and MM hearts treated by paraquat are shown here. You can appreciate that many of the MM hearts stopped beating after paraquat treatment, while most of the VV hearts stayed active. And we quantified the dysfunctional hearts in this graph, showing significantly more dysfunctional MM hearts. These results suggest that CaMKII oxidation by pathological levels of ROS can contribute to frailty, in part by acting in the muscles. Our findings suggest that CaMKII oxidation mediates ROS hormesis. With the physiological level of ROS, CaMKII activity is optimal, and it promotes physiological performance. While ROS level is increased either by paraquat treatment, by diseases, or by aging, hyperactivation of CaMKII leads to reduced performance, frailty, and aging-associated diseases, and death. We also show that taking away CaMKII oxidation, either by genetic manipulation, by introducing the VV residues, or by antioxidant, abolished this hormesis effect. To summarize, using 2 model organisms, we established that CaMKII oxidation is an evolutionary trade-off that confers performance benefits, but also increases susceptibility to aging-related diseases. CaMKII oxidation is a novel molecular example of the antagonistic pleiotropy theory of aging. Additionally, we showed that CaMKII oxidation provides ROS with an amino to confer the hormesis phenomenon, where too little or too much ROS lead to adverse outcomes. So what's next? Our ongoing project is built on the data I presented here. In short, we will use the animal models to identify upstream regulators and downstream targets of CaMKII oxidation, using proximity labeling and RNA sequencing approaches. We will also explore the roles of CaMKII oxidation in aging with human samples and primary cells. The goal is to identify potential ways that can preserve the beneficial roles of CaMKII oxidation while preventing its detriments, which may help us to achieve healthier aging. So in addition to the collaborators I highlighted in the talk, I'd like to make additional acknowledgements. first and foremost, I'd like to thank Dr. Mark Anderson for supporting me in developing this story that has a quite different flavor than our lab's direction of research. And furthermore, this project was supported by our current and former lab members, even though some of them are not directly involved in it. I'd like to thank you for your attention.
Thank you very much. Kichuan, this is a provocative talk.
Thank you.
So very, very interesting. So that would mean that if we, by using CRISPR to replace the MM residues for the BB residues, it could prolong our lives in the future?
That's an interesting possibility, but I think what my study mainly tells us is maybe the role of MM residues are so nuanced. So it's possible that the MM residues play predominantly detrimental roles in the context of aging. If that's the case, eliminating the MM residues and replace it with VV would be beneficial. But I think more likely, MM residues of physiological functions would not magically disappear in the context of aging. So more likely than not, MM residues are still physiologically important. So probably what we want to do is not to eliminate it completely, because if we do that, we may lose the ability to, say, fight infection or to repair injuries. Instead, we may need to find regulators or targets that can be temporarily manipulated without blocking the beneficial effect of MM residues.
Okay, thank you.
Mm-hmm.
In the same line as Alicia, Quinchana, before I go into the questions from the audience, I wanted to ask you if we had a time machine where we could travel millions of years ahead, and thinking of our sedentary life, which is growing and growing, would you predict that we would lose the oxidative capacity as we would no longer need it? Because what do we need extra muscle function for if we're having everyday life a more sedentary way of life?
I don't know. I think being athletic might be attractive in finding a mate and propagate the genes. So that's just my joke. But I think CaMKII oxidation likely have many rules. So we used motor function as an example, because it was the easiest function to tap in, and it also improved motor function was one of the features that appeared in the early vertebrate evolution. So it was obvious to us at that time when we did this study that it might be one of the functions involved in CaMKII oxidation. But as you can see from my presentation, we also found that CaMKII oxidation is involved in mast cell degranulation and involved in gene transcriptional regulation. And these are only two CaMKII we looked at. And there are CaMKII gamma, CaMKII alpha and beta. They also are oxidizable. So we think that CaMKII oxidation likely will be here for a long time because they have many rules. And actually, I searched genome databases. I only find like one case where the MM residues have a SNP in one single individual. So a mutation of the MM residues are probably not very well tolerated.
Okay, thank you, Chang. So I have some questions from the audience, but before I want to acknowledge Mark Anderson because he was going to actually co-chair this session, but he unfortunately had some family problems, so I stepped in for him. But we would like to thank him and hope that he resolves his family issues shortly. So
thank you.
Questions from the audience. So Dr. Palomeque asks, it appears that oglycogenation of CaMKII 2 is previous than oxidation in the evolution. Does that mean that oxidation is more significant than glycosination for the deleterious effects of CaMKII?
That's a great question. We don't know about that yet. So O-glycosylation is involved in transducing pathological signals in diabetes. So it also can have detrimental roles. And we think that, so actually I can go back to my slide. So you can see the first post-translational modifications evolved was the phosphorylation of the threonine 287. This is a well-known site. So it actually evolved together with the ancestral vertebrate, ancestral CaMKII about 1.2 billion years ago. So we think that it's involved in the most fundamental function of CaMKII, probably determining most of CaMKII's properties. And those others are the add-on functions. But the sugar modification on serine 280 evolved also quite early. So we think that it plays significant physiological functions. Probably it has more relationship to metabolism because the levels of glucose in the cells is probably an upstream signal for it. And then CaMKII oxidation is probably evolved as an add-on to improve motor function, but later it's propagated into all other CaMKII isoforms. So we think that expands its evolutionary role in other animals as well. As for the detrimental functions, I think it will be case by case. As the antagonistic pleiotropy theory suggests that, and the hormesis phenomenon both suggest that a good thing can be good only when it's at proper level. Too much or too little are no good. So I think all of them have the potential to be bad.
Okay. Another question from Luis Gonano. He enjoyed very much your data. How do you separate the relevance of skeletal muscle function and heart function in the treadmill performance?
That's a great question. So I didn't show the more detailed experiments we performed. So we first did a treadmill experiments and found a reduced motor performance by the knock-in mice. And we asked exactly the same question you asked. How do we separate cardiopulmonary input or even nervous input that regulate skeletal muscle function? So what we did was 2 layers of experiments. We first used the in situ contraction experiment. Let me see here. So we anesthetized the mice and we fixed the mice on this apparatus. And we directly stimulated the nerve to induce quadricep contraction. And we measured the force output for fatigue. So this isolates in large part the input from the nervous system and potentially in large part from the cardiopulmonary system because only a group of muscle is involved. And we further isolated muscle fibers from the mice and did in cellular experiment. So this experiment was done with isolated muscle fibers. And we observed an effect of blocking CaMKII oxidation on calcium handling. So this obviously isolated the other confounding factors. So we think there's an intrinsic role for OX-CaMKII to play in the muscle fibers. However, our study didn't exclude the contributions from other tissues. And it's very likely other tissues such as tissues, the liver or even the adipose tissue also contributes to the motor performance phenotype because they can provide substrates for exercise. So that will await future studies.
Okay. Alicia, I think we have time for one more question. Do you want to ask another question?
I would like to know whether these experiments, just the one that you are showing here, exclude the possibility that the phosphorylation of CaMKII is playing a role.
Would you please repeat that again? I didn't hear that. The volume is kind of small, low.
Whether the phosphorylation of CaMKII is playing a role in this increase in tetanic force, this increase in calcium.
Oh,
Because you are to get the tetanic force, you have to increase the frequency of the stimulation, and then there will be an increase in CaMKII phosphorylation. It is possible to exclude the role of CaMKII phosphorylation here.
no. Actually, what I'm thinking is, Dr. Matias's question was whether CaMKII phosphorylation also contributes to this phenotype in our experiments. And my answer is we cannot exclude the role of T287 phosphorylation in this experiment. Actually, I think the post-translational modifications in CaMKII regulatory domain are likely interacting with each other. So CaMKII oxidation, as recently shown by a paper in JMCC by Dunbar's lab, actually oxidation contributes to calcium calmodulin binding to CaMKII. It increases the binding and prevents dissociation. And calcium calmodulin binding under sub-maximal calcium level can happen when CaMKII is oxidized. So I think that some of the effects, such as here, reduced CaMKII activity due to removing the MM residues and replacing it with VV, could happen in part because the phosphorylation is also reduced.
Okay. Okay, thank you. Thank you very much.
Thank you so much.
Okay, thank you very much, Rinjan. We really enjoyed your talk. So we have many new things to look at and to think about. Now we know all these aspects of CaMKII too. So thank you very much for opening our eyes to all this new field.
Thank you for this great opportunity.
Okay.
Okay.